Hello, everyone. I'll be talking about interplay between nucleosome dynamics and histone modification in epigenetic inheritance based on a recent model that I'm working on. To begin with, our DNA is very long, which is present in our cells by being wrapped around proteins called histones, forming structures called nucleosomes. These nucleosomes show two very important properties. First, they are very dynamic. They can bind, they can unbind, they can slide. And in addition, they can communicate to each other. They can literally talk to each other with the help of certain modification marks, which is present on them. Essentially, these modification marks are encoded as a pattern in the cell. So the question we are interested in is how do cells maintain and inherit the pattern? Based on the recent experiment done by Grewal's lab, the picture shows what they suggest. So what we have here is, each sphere here is the histone protein wrapped by DNA, forming nucleosomes, and each nucleosome has got the acetyl mark represented by the green triangles. These green triangles are removed by enzymes called as histone deacetylase. This removal of green marks actually doesn't let the remodeler named SwissSNF to act on. This is because the SwissSNF remodeler has got a domain in it known as bromodomain, which is responsible to recognize the acetyl patches. So once the acetyl marks are absent, SwissSNF remodeler cannot recognize and it cannot perform its function, which is known to be nucleosome eviction and nucleosome sliding. In addition, the absence of these green marks actually allows the histone methylase, denoted by CLAV4 here, to actually put methyl marks on the nucleosome. The opposite thing happens when we don't have histone deacetylase enzyme. So in this case, the acetyl marks cannot be removed. The SwissSNF remodeler can act on the nucleosome and can remove them. We wanted to model such a phenomena, and the question we asked is, can existing model help? The answer is no. This is because almost all the existing models present in the literature either focus on histone modification spreading kinetics or on nucleosome dynamics. None of the models actually consider both. So here comes the need of a model to actually account for the following. The first is nucleosome binding. Given a bare DNA, nucleosome can bind. Modification dependent disassembly. With respect to the presence of acetyl and methyl mark, it can disassemble faster or slower. Similarly, sliding, where the presence of the type of modification mark dictate whether the nucleosome can slide faster or slower. Deacetylation, where the acetyl mark is removed, and methylation spreading, where the methyl mark is spread to the neighboring nucleosome. So here is what we propose. Any nucleosome binding will bind with an intrinsic rate R on, and the nucleosome will be acetylated. The acetylated nucleosome can get deacetylated with an intrinsic rate RDAC. The deacetylated nucleosomes can get methylated condition that the immediate left neighbor is methylated and the distance between the two, D, is less than the size of the spreading enzyme, ES. Acetylated, deacetylated, and methylated nucleosome can unbind and slide at rates R of AMU and RSAMU respectively. At the left barrier, we do have a nucleation point, which will act as a methylation hub to initiate the spreading. So we varied different model parameters to check the effect. The first thing we did is we switched off spreading and varied the rate of deacetylation. What we find is increasing the rate of deacetylation increases the nucleosomal density. And this is because more the deacetylation, more will be the number of deacetylated nucleosomes. And the deacetylated nucleosomes have got lesser rate of eviction. The next thing we did is we switched off deacetylation and we went on wearing spreading to check its effect. What we find is in absence of deacetylation, spreading cannot make any difference. And this is because only the deacetylated nucleosomes can get methylated, and therefore there has to be some amount of deacetylation happening to make spreading make the difference. So we included deacetylation, and what we find in this case is when we increased spreading, the nucleosomal density also increases. This is again because more the spreading, more will be the chances of finding methylated nucleosomes, and methylated nucleosomes have lesser chances of eviction. We then varied the dissociation rate itself of nucleosomes, and what we find is when the rate of dissociation is very fast, spreading cannot make any difference. This is because before the spreading could actually happen, the nucleosomes tend to dissociate. So we then ask the question is, under this condition when disassembly of nucleosomes are very fast, is it so that spreading can never make the difference, or any way spreading can be made to make the difference? The answer is yes, it can be made, provided we selectively lower down the removal rate of methylated nucleosome. So this essentially increases the time for which the methylated nucleosome would stay, and therefore the spreading becomes effective. Another thing that our model has allowed us is to profile methylated nucleosomes at steady state. So the graph shown here actually depicts, given a lattice site, say 0, what is the probability of that site being covered by a methylated nucleosome. So we do find two things. First, as we move away from the nucleation point, the methylation profile show a decay. And the second thing, the decaying in this methylation profile can actually be regulated by regulating nucleosome dynamics. So in case when there is slower disassembly, we do find very strong statistical positioning of methylated nucleosomes, which are in some way or the other an indication of strong boundary. But in case of faster disassembly, the methylation gets restricted to certain domain. Again, the strong statistical positioning reappears once we selectively slow down the removal rate of methylated nucleosomes. We then actually switched off all the nucleosome dynamics part to check what exactly can modification kinetics do. And what we find is having this nucleosome dynamics is essential to get the statistical positioning of nucleosomes, which cannot be get provided we don't have the nucleosome dynamics and only modification kinetics. Coupling nucleosome dynamics along with histone modification kinetics has also allowed the restoration of histone modification marks irrespective of the initial density we start with, which is obviously not possible in absence of nucleosome dynamics. So here's what I have as a summary. Slow disassembly along with deacetylation and spreading are collectively responsible to increase the nucleosomal density. Methylation profiles show a decaying oscillation, which is an indication of strong boundary and can be regulated by regulating nucleosome dynamics. Modification kinetics alone is not sufficient enough to restore the histone modification marks. Although our model was able to beautifully establish the relationship between modification dynamics and nucleosome dynamics, we are still striving to actually understand if this is the sole secret to make epigenetic inheritance robust. If yes, is this the only secret, or there are many more secrets waiting to get out? So with this, I'd like to thank my supervisor, Professor Ranjit, my lab, and everybody present here. And I'm open to questions. Thank you.
Thank you. Any questions?
Sorry, I missed something basic now. When you're talking about epigenetic inheritance, are you talking about the transmission of the modifications from, I mean, you're not talking about the transmission of the modification from generation to generation.
No, I'm talking about histone modifications. histone modification mostly, yeah.
Seems to me that the rules that you have proposed are all equilibrium rules. So, right, and I was wondering, but despite, and you also have some spatial dependence. You assuming some underlying diffusion.
Not exactly underlying diffusion as
So where does
such.
space come in?
So,
Yeah.
Yeah.
You have a sliding term, isn't it?
correct, yes.
Okay. So that's where space comes in.
Yeah.
But then everything should be, I mean, it should be analytically solvable, no? It's all equilibrium plus a translation.
Yeah, so basically, we actually follow kinetic Monte Carlo step. So once we initiate from any configuration, we let the system evolve for a long time. So and then in this way, we actually let the system be in equilibrium.
But I'm sure you can solve it analytically.
Hi, nice talk. So I just had a few doubts. Like when you're talking about inheritance, do you mean imprinting in the next generation or just inheritance like from one cell to the next cell once it divides?
So it's from one cell to the next.
Okay, so you're not dealing with imprinting.
Okay.
So it's like each time step is one Monte Carlo time step. So it's like to complete one event, the time it takes. Probabilistic time it's take to get done with one event. Yeah, so mostly what is known till now is RON takes about 12 per second. So if we wait for 1 second, we find 12 nucleosomes binding. So with that, we varied other factors. So that's it.
Okay. If there are no other questions, let's thank Sai again. Thank you. Next speaker is from my lab only, from IIC, Miki Anand.